Hello and welcome to today's webinar. On behalf of European Pharmaceutical Review and Nelson Labs, I would like to thank you all for attending. My name is Catherine Eckford and I'm an editorial assistant of European Pharmaceutical Review, and I will be moderating the audience Q&A at the end of this webinar. Joining me today is Peter Cornelius, Senior Microbiology Expert at Nelson Labs Europe, who will be discussing how to control endotoxin contamination during pharmaceutical production. Following today's presentation and discussion, we will move on to a live question and answer session where you can pose questions to today's speaker. Please remember you can submit your questions at any point during the webinar using the questions panel situated in the menu on the right. Now I would like to hand over to Peter, who will begin our webinar today.
Yes, hello everybody. My name is Peter Cornelius and I work for Nelson Labs, and thank you for attending our webinar today about controlling endotoxin contamination. Just a few words about our company. So I work for Nelson Labs. I am based in our Belgian facility in Europe, and we are part of the Sotera Health group, and amongst others, our sister company is Nelson Labs is specialized in contract services for pharmaceutical and medical device industry. Sterigenics, you all probably know, is world leader in sterilization services, and then Nordion, our third sister company, is specialized in the creation of manufacturing of radioisotopes. So today the topic is about endotoxin control in pharmaceutical production, and I'm going to start with some sort of conclusion of this webinar by stating that endotoxin control is not obtained by just one moment, I'm sorry. So endotoxin control is not obtained by end product testing alone. It's a little like aseptic manufacturing, where also just testing your end product does not give you guarantee about the absence of a certain parameter. So if you want to control endotoxin, what you really have to do is to study your production process and perform risk assessments. But of course, to do so, you need to know about the principles of endotoxins. What are endotoxins? Where are they coming from? How can they possibly be removed? And of course, how can if they are present, how can we detect them? And what are the pitfalls? So the first part of my presentation will, in a very short manner, be about endotoxins, the origin and characterization, and how we can detect them. And the largest part of my webinar is we are going to use those principles and see how we can use them to perform a risk assessment and to perform endotoxin control. So endotoxins. Endotoxins are a pyrogen, which means they elicit a fever when you inject them in your bloodstream. Endotoxins are not the only pyrogens, so you have a number of pyrogens, but it's the most studied ones, and it's the most potent and the most occurring and the most resistant of all known pyrogens. And as you probably already know, endotoxins, or another name is lipopolysaccharide, they come from the outer cell wall of gram-negative bacteria. So the gram-negative bacteria has a double cell wall. On the picture below, you can see a picture of a gram-positive cell wall and a picture of a gram-negative. Just for your information, teichoic acid or peptidoglycan, which occurs with both organisms, they are also known pyrogens. But the endotoxins, well, they reside on the outer membrane, and the active part is actually inside the membrane of the bacteria, and that's why we call them endotoxins and not exotoxins. And as I told you, they are fever-inducing when you inject them in a bloodstream of a patient. When we look into more detail in the structure of endotoxins, you can see that it has a polar part and it has an apolar part. And the active part is the part with the fatty acid, which is called lipid A. They are highly negatively charged. And why is that so important to understand this? Is that they stick very well to positively charged surfaces like glass. So if you have pyrogens and you inject them, or you into a glass vial, or you have an infusion bag, also has the same problem. Well, they most probably will stick very rough on the surface of the container. Yeah, and one of the first studies that I did when I started working at the company 20 years ago was we tried to inject or test filters for endotoxins. We injected the filter, the IV bags, and we didn't find anything back, just because they were sticking on the surface of the polymer bags. They are very resistant to high temperatures, and that's also a very important feature, because if you will sterilize your product by either ETO or using steam, they will not be destroyed. So steam sterilization or ETO, they will destroy destroy the bacteria, but you will get a release of endotoxins, because the cell wall is completely destroyed, but the endotoxins are left untouched. To get rid of endotoxins, the only thing that you can do if you're using heat is to heat at least 30 minutes at 250 degrees Celsius. But they are not resistant to high or low pH. So if you would clean with a solution of a molarity below 0. Or above 0.1, you will destroy the endotoxins. Since they have a polar and an apolar part, they react as surfactants when you add them to water, and depending on the fatty acids, where they are branched and how they are branched, they will form different structures. They can either form lamellar structures in water, or cubic, or hexagonal. And why this is so important, and we'll get back to this later, is because to be active, endotoxins need to be aggregated, and depending on how they are aggregated, it determines their activity. For an example, Rhodobacter, it has lamellar, it forms a lamellar aggregate, and it is very inactive. And one of the most active LPS or endotoxins are the endotoxins of E. coli, and they form hexagonal structures. So this is important to understand, that not each species of gram-negative bacteria will have the same activity for the endotoxins. So a small wrap-up of this first part. So what is the relation between bioburden and endotoxins? If you have bioburden, or if you had bioburden, you destroy the bioburden, and if there were gram-negative bacteria in it, you most probably will have contamination with endotoxins. And also important to know is that gram-negative bacteria, they tend to grow very well in water systems. And a lot of our customers that having issues with endotoxin contamination, the first thing that we ask is, what about your water system? A lot of examples that we see, the endotoxin contamination comes from the water system. If you sterilize a product, being it a glass vial or being it your base material, if you sterilize with ETO, gamma, or steam, it will not affect or not destroy endotoxins. There is a small effect. We performed a study 2 years ago on the effect of gamma irradiation on endotoxins, and gamma irradiation does have a small effect. What we noticed for most bacteria is that we got a 10-fold reduction. So it's not enough to claim that it's depyrogenated, so it does not destroy or render an endotoxin-free product, but it does have a small effect. Most disinfectants also, it will kill bacteria, but it will not destroy endotoxins. And then finally, if you perform contamination control, you will not get rid of endotoxins. But of course, if you control your bioburden, if you do not allow gram-negative bacteria to grow, you will not have a rise in endotoxins. So contamination control has an effect on the endotoxin control. And then a few words about the endotoxin detection method. I'm going to be very brief about this. There are a number of endotoxin test methods available. but I'm only going to treat the LAL assay, since this is the one that is most frequently used. The LAL assay is based on the blood of this fossil or living fossil. It's the Limulus polyphemus, or the horseshoe crab. And it has this animal has a very primitive immune system. When it's being infested by gram-negative bacteria, blood forms a clot, and it prevents the infection from spreading throughout the body of the crab. And endotoxins, they activate what is called factor C. So factor C is one component from the cascade of this reaction. Factor C will activate factor B, and this will eventually activate a pro-clotting enzyme. And for the chromogenic method, which we in our lab use most, it activates a chromogenic enzyme, or it can be a turbidimetic enzyme. Just a quick word about the recombinant factor C assay. This is, well, it's not new, but its approval is quite new. From 2020, it's part of the EP. There is already at FDA. It's not in the US Pharmacopoeia, but there is since 2018. It is being used for batch release in the US. Recombinant factor C is an enzyme that is produced in vitro, so you do not need the horseshoe crab anymore. So it's a more sustainable alternative than using the general LAL method. And because it's produced in vitro, it should have less lot-to-lot variability, because it's a well-known fact, since you're using a product from animal origin. It can be dependent on from which organism it came. So it's a well-known fact when using LALs that you have a lot of variability between different batches. Test interference. So if you perform endotoxin test using the LAL assays, there are a number of things that you need to keep in mind. You can have false negatives, which are either caused by inhibition or endotoxin masking, and you can have false positives, which can be by enhancement of the test enzyme or by the presence of beta glucans. Inhibition and enhancement is a traditional test as an assay suitability. And if we look at what's in the pharmacopoeia, this is what you have to do to assess this. So if we are talking about assay suitability of an LAL assay, we have to test your solution neat without adding something. We have, which is in this table, solution A. We test your test item or your product by adding endotoxins that are equal at the middle concentration of our standard curve. We have to include at least 3 concentrations of endotoxins diluted in water. So that's actually our dilution series, and we have to test the water. So if we look at this is actually included in each and every single assay that we perform in the lab. So I always answer to customers that come to us with the question, I have to validate my assay. Well, you can do it. It should not be a separate assay. You can do this, but actually this is what we do in each and every assay. So each test that we do validates its own result. I often get questions about, well, shouldn't this be performed on 3 batches and different dilution? Well, this is actually something that comes from an FDA guidance, which was, well, the FDA guidance was from 1987, but since almost more than 10 years, this guidance has been withdrawn. So this is not true. So you do not have to perform this on 3 batches. The only thing that EP or USP tells you to do is what's in that table above. And then there's another test interference which is a little, well, it's much more complex, which we call low endotoxin recovery, or another name that you probably would have heard is endotoxin masking. And this is a time-dependent decrease of endotoxin activity in your sample. So it means that if you add or if you have endotoxins in your product, you test it immediately after addition of the endotoxins. You will find them in your assay. If you wait like 24 or even 48 hours, it might happen that you won't see any activity at all. And this is something I think you can test very easily in the lab, and which we have seen for quite a number of products, that if we add endotoxins for like 50 EU per milliliter, that if we wait 24 hours, you won't find any endotoxin at all. And this is different from inhibition and enhancement, because it's actually a destruction of the endotoxin aggregates in the sample. And when you look at what inhibition is actually any substance that might interfere with the proper working of the test enzyme, the factor C. And so the only thing you can demonstrate the presence of this, you won't notice this if you just perform inhibition and enhancement testing. The only way to demonstrate that you have endotoxin masking is to organize specific hold time studies. So that's nothing more than adding endotoxins and testing at the different time points how many you will retrieve. Just to let you understand what endotoxin masking really is. So I told you that you have to have some aggregates for the endotoxins to be active. And if you have a chelator like citrate in your product and a surfactant like polysorbate, the first thing that, well, these aggregates, they are stabilized by divalent cations. And if you have a chelator present, it will capture that divalent cations and destabilize the aggregates of the endotoxins. And then subsequently, if you have surfactant, it will completely destroy the aggregates. And of course, that results, as I told you, if we no longer have endotoxin aggregates, you won't have any, or you won't find any endotoxin activity in your assay. And then the last interference that we notice, and you should very be aware of, is the presence of beta-glucans. So if you look at how the reaction cascade goes for LAL, you see that if you have beta-glucans, there is also factor G present in the lysate of the horseshoe crab's blood. And beta-glucans, they will activate factor G, and this also will activate the clotting enzyme. So to overcome this, and this, well, beta-glucans, just to let you to know where they can come from, if you use cellulose as a filter, well, cellulose is a beta-glucan, so you can have beta-glucan contamination, and this might result in a false positive result. If you suspect this, you should add, well, there are a number of possibilities that you can, well, there are actually two possibilities that you have. First of all, if you can use beta-glucan blocker, that's a product that blocks the factor G pathway. And another option that you have, as I showed you, is recombinant factor C, because it does not activate pro-clotting enzyme. So the factor C immediately activates a fluorogenic enzyme. So factor G and beta-glucans will not have an influence on this assay. Okay, and then for the biggest part of the major part of our of this webinar is the endotoxin control. We're now going to use these principles to perform risk assessment and to look where we can intervene in our production process, what we should control to have a final product that is endotoxin free. I wanted to start with a statement which is published by FDA in Questions and Answers in 2012, and this actually is telling you what to do. So if you have a production, if you produce either pharmaceuticals, but the same principles are applicable for medical devices, you should look into every aspect of the manufacturing design. The impact of process holding time, endotoxin removal steps, etc., etc. So it's not only, as I said, it's not only end product testing. So why not just test the final product? Well, since you probably know, if you have bioburden control, if you have, if you're performing aseptic manufacturing, the bacteria, they will not be present on, well, not necessarily be present on every single device or every product. So if you just to understand, let you understand, this is a statistical example. If you're actually wanting to prove the absence of something, and the absence of something, proving the absence of something scientifically can only be done by testing every sample, and that's of course something you don't want to do. And just to show you what the impact is of testing a few samples of a large batch, if we would have a batch of 50,000 units and we would have a contamination of 1 in 1,000, and you would test only 3 units, so that's normally the average that we test for pharmaceuticals. We test 1 at the beginning of the batch, 1 at the end, and 1 in the middle. We have a 0.3% chance of getting a positive test. So you get actually very little information from end product testing. So the only thing that you can do is to guarantee the absence of endotoxins, to monitor the process, avoiding too much entry of endotoxins, so performing risk analysis, where can the endotoxins come into the chain and where can I control them, how can I limit them. And that's, well, there are a number of risk assessment methods, but the ones we are handling today is called an HACCP study. And this is short for hazard analysis and critical control point. A critical control point, very easily to understand, is a place where endotoxins contamination might enter or might be removed from your production chain. And to perform these types of analysis, you have to understand the microbiological principles. So that's why I wanted to start with what are endotoxins and how can they be tested. You have to understand the endotoxin properties. And of course, without saying, you have to understand your production process. And since this requires so many expertise, to perform this hazard analysis, you need your team of experts. You need your QA, engineering, maintenance, QC, microbiology, validation, regulatory, and development. So this is not a one-man job. There are 7 steps in an HACCP analysis. And we are mainly going to focus on the first 3. So conduct hazard analysis, identify the CCP, and then assigning limits for each CCP. Those are the 3 most complex. The other 4 are actually quite logical. And the way I wanted to present this to you is to apply these principles on a generic production process. So we have a production process that starts with testing the receipt and testing of raw materials. Then in a non-sterile environment, we are going to formulate our drug, which consists out of saline, dextrose, an API, and water for injection. And the dextrose is present in the highest concentration of 150 milligrams per milliliter. So that's a lot more than just saline. That's something that will be important later on in my explanation. And then before entering the sterile environment, we are going to run everything through a filter of 0.22 micrometers, perform ultrafiltration to remove endotoxins at a certain up to a certain level, and that's being performed in a sterile environment. Subsequently, we are going to fill our product in a vial and close, again in a grade A environment. And finally, we have our finished products that we will subject to endotoxin testing. So the first step in an HACCP analysis is to conduct a hazard analysis. And the hazard today is endotoxin contamination. And of course, if you have endotoxin contamination in your product and you administer this to your patient, they can have fever reactions, organ failure, or even the death. And important to understand with the explanation today is we are studying endotoxin contamination. We are not studying the possibility of having a non-sterile sample. So that's something you can use the same principles of HACCP for aseptic manufacturing, but then of course the hazard is a non-sterile sample and not endotoxin concentration. And that's important to understand, and I'll show you in a minute why this is so important. Second step is to identify your critical control points. And to do so, you can use this decision tree. The first question that you need to, so what you need to do is you have to break down your production process. And for each individual step, you have to run through these different questions. So does this step involve an hazard or significant risk or severity to warrant its control? If the answer is yes, you should investigate if you have preventive measures to perform some control that this possible entrance of endotoxin does not go through the roof. If the answer is yes, you have preventive measures, you should ask yourself the question, is it now necessary that I control this step to eliminate or reduce the hazard of this endotoxin concentration? And if also the answer to this question is yes, then this is identified as a critical control point. Let me explain this too by running this through our example. So the first step in our production process is the receipt and testing of our raw materials and our API. And there are some assumptions that you can make and that you must make before starting this analysis. First of all, materials from natural sources, well, it's a well-known fact that these are potential, they have a high likelihood for endotoxin contamination. On the other hand, inorganic salts or chemicals or strong acids and bases, they normally do not contain a lot of endotoxins. Those are the first assumptions that you can make in this risk assessment to answer the questions from the decision tree. Of course, those are assumptions, and you really need to test this before you start your risk assessment, but that gives you a good baseline. If now we use these assumptions and we are going to well, I'm going to show you only for sodium chloride and dextrose because they are very much different. If you pose yourself the first question from our decision tree, first of all, sodium chloride is an inorganic salt, so probably it will not contain so much endotoxins. And what you should do then is test a few batches of your saline, and you will find out, well, it does, that this assumption is true. So does this step involve hazard or significant risk or severity to warrant its control? Well, the answer to this question is no, so it's not a critical control point. For dextrose, on the other hand, dextrose comes from a natural source, and let's assume that we tested our dextrose and we revealed that we have quite a considerable lot-to-lot variability. So the answer to this question is yes, and we go up to the next question: does a preventive measure or hazard exist at this step? Well, the answer would be yes, since I assume, or in this example, we have a vendor that has a COA available. We have performed vendor qualification, and we have some level of comfort about the endotoxins that we will be introducing into our production chain. So the answer to this question is yes, we have a preventive measure. So we shift to the last question: is control at this step necessary to prevent, eliminate, or reduce the hazard? Well, dextrose, as I pointed out, is a huge formulation component of our final drug. So we might feel more comfortable if we would test each lot, so we are really sure and really aware of how many endotoxins that we are actually introducing in our chain. So the answer to this question is yes, and we have by this identified a critical control point. One more example in our production chain is the holds step. So before we go into sterile filtration, our pharmaceutical formulation is being kept for a certain time at a known temperature in the production area before we actually can aseptically filter it. So holding a non-sterile sample, of course, it's not so difficult to understand that this is prone to bacterial growth. So does this step involve a hazard or significant risk? Yes. When there are gram-negative bacteria present and you have a lot of humidity or possible, well, we have in our product, we have water for injection. that is actually a good base for letting gram-negative bacteria grow. So this is the answer to this first question is yes. Does a preventive measure for the hazard exist at this step? Yes, because, well, in our case, and that's quite logical to do, we have to validate this holding time. How long can we hold? At which temperature can we hold to have the bacterial growth under control? So we have a preventive measure. And then the last question, is control at this step necessary? Yes, for sure. Because if we have a lot of gram-negative bacteria on our raw materials, we might have a large contamination after this holding time, and the contamination can perhaps go so high that it will exceed the deporiginating ability of our downstream processing. So we have identified a second critical control point, and if you do this on every step of your production chain, you might come up with these 5 critical control points. And something I want to point out, when we started the HACCP analysis, I pointed out that the first step is to identify your hazard. If you look, for instance, to the filtration through a 0.22 micron filter, this is certainly a critical step for your microbial contamination. So I'm not saying that this is not a critical control point, but it's not a critical control point for endotoxin contamination. It's a CCP for bacterial contamination. So it's very important to identify your risk and to stick to your risk, to your hazard. And then the third step in the HACCP analysis is to assign limits for each CCP. Again, we're starting with our complete production process, and a good point to start is, well, it's actually the only point where you can start, is start where you can find limits in pharmacopoeia. And for finished product testing, that's what you can. That's something what you have. If you know, that's probably not new to most of you. If you know the dose of your product, you know the average weight of your of a patient, you can calculate the limit of your product. So what we're now going to do, we are going to start at the end of our production chain, and we are going to more or less distribute this allowable endotoxins among all the critical control points in our chain. So this is a simplified production chain with only the CCPs. Let's say, for instance, then we have a limit of less than 350 EU per milliliter for our finished product testing. The first step that we encounter, our critical control point on the way back, is the deperogenation of the components. Are we going to test these glass vials and stoppers at the end or before the deperogenation of our material? Well, that's probably not a good idea. We have validated the deperogenation step. It gives us at least a log 3 reduction. Again, if you want to prove the absence of endotoxins, you won't get so much information by performing endotoxin tests on deperogenated material. And also, most glass vials and stoppers, they will not contain so much endotoxins that the log 3 reduction might not be enough. So a better point of checking this critical control point might be to monitor time and temperature of the deperogenation oven. So we're not going to perform endotoxin testing on these components. The second critical control point is immediately after the ultrafiltration step. It might sound logical to have the same endotoxin limits, since we do not encounter any possible contamination, or we didn't imagine them during the next steps. It might sound logical to set the same limits as the end product testing, but we want to feel more comfortable that if something might happen, we're still within our specifications. And if we would set a limit of 175 EU per milliliter, we might always reach the end product specification of 350. One thing that I should tell you, this is not an exact science. So we are starting with figures that might sound reasonable, but as you go along and you learn to understand your production process, it's a dynamic issue. So we might see that these limits are not obtainable, then we have to adapt them. We might see that we can loosen them. That's something that you will learn along the way. The next critical control point is the hold step. There we have to keep in mind, or to the history of the formulated bulk, we have to keep in mind the efficiency of our ultrafiltration process, which is of course something that you validate. You know exactly how much, or at least how much endotoxins you will be able to remove by ultrafiltration. And we use a validation data from the hold time studies. And by doing so, we know that the ultrafiltration will at least give us a factor 3, so a log 3 reduction of the endotoxins. So if we would allow a factor 10 difference compared to the previous limit of 175 EU, we are always sure that if we have, after the hold time of the product, if we have less than 1,750 EU per milliliter, we will always reach that 175. So we build in quite a lot of safety margins that we are always sure that we control, and we will not exceed the limits farther down the production chain. If we look at this a little bit closer, of course, we will also have a batch review of the actual hold time. So this is actually a second control for this critical control point. So we will look at batch at the hold time and temperature in our production chain, and this should be below the validated values. And then finally, this is a little more complex than the other, setting the other limits. We look at our raw materials. We have 4 different raw materials. We use the same limit for the total product of 175. Of course, not the 1,075 EU per milliliter, because these endotoxins, they might might originate not because of the endotoxins that are present in the beginning of the hold time, but simply by growing of bacteria during the holding of our product. So we use for the APIs 175 EU per milliliter for the total formulation. And we are now going to distribute this throughout the different raw materials. And if you do not think this thing through properly, you might just distribute them equally to all components. But that if you would count that, if you would calculate that up to the how much per milligram of product you can have, you might see that, well, in this case, it means that our most strict specification is actually on dextrose, which, as we demonstrated, has also the highest possibility of having an endotoxin present. So this might not be the best idea. And certainly also look at water for injection. If we would do this, if we would equally distribute over all components, we would have a limit for 43 EU per milliliter for water for injection. And of course, you all know probably the limit for water for injection, which is 0.25 EU per milliliter. So that's a little, that sounds unlogical. So what a better suggestion is to take the likelihood of contamination and the relative amount into account. We know dextrose is present in the highest concentration. We know that dextrose is prone to the highest possibility of finding endotoxin contamination. So let's set 90% of our endotoxin, of our allowable endotoxin, to dextrose. Then we find a limit of 1.05 EU per milligram of dextrose. And saline has then, of course, a very low specification, the same for the API. And since water has such a low, water for injection has already guaranteed such a low amount of endotoxins, we are going to ignore the water for injection. So by doing all this, we find limits for all our critical control points. And so these 3 steps are the most complex ones. The 4 last steps are relatively easy and relatively straightforward. So we now have our limits. The next thing is we have to develop validated tests and verify if what we said would be a limit is actually obtainable. So we now start to develop tests and starting to test at our critical control points. The next step is to verify corrective and preventive actions. This is a good thing to do before you're actually starting your production, because if something might happen and you've thought very carefully about all the possible things that can go wrong, you can upfront already set up corrective and preventive action, and this will allow you, when something happens, to react faster and sooner. The next step, verifying operation procedures and critical control points. So you now have to establish how frequent are you going to monitor, who is going to monitor, who do I need to train for this, what are my preventive maintenance steps, and how I am and when I am going to calibrate. And then, of course, that goes without saying, verify that all records of each control point are documented. And that's everything I have to tell you about the risk assessment. So to conclude my presentation, what are the lessons that we've learned? So very importantly, just like aseptic manufacturing, you cannot just rely on end product testing to guarantee that your product is endotoxin free. You have to perform a risk analysis and control and imply control measures. And the way you can do this is by performing a risk assessment according to HACCP. To do so, you really need to understand your processes. So you require a lot of, well, you require a number of experts over a number of domains to help you with this. You have to understand what introduces the endotoxins, and you have to understand what removes endotoxins. You have to build in at all critical control points, you have to build in safety margins to make sure that you always obtain your limit for your end product. And important to understand, this is a dynamic product. So when you go along your path, you can adapt your values, you can adapt your limits. You might even learn that some points that you thought were critical control points might not at all have a huge impact on the results. So this should be a continuous process of evaluating and understanding the risk of your production process. And by this, I want to thank you for your attention. If you have any questions that would not be answered today, feel free to send me an email. You can see my email, my address down below. And finally, my last slide. This webinar, amongst a lot of other scientific publications, you can find at the Sotera Health Academy. Just register. The admittance is free, so feel free to look for more information. Thank you.
Thank you, Peter. That was a great presentation. Now we'll move on to our question and answer session. Please don't forget you can still submit your questions to our speaker by using the questions panel in the menu on the right, and we'll try and get through them all. Now let's take a look at the first question.
So 3 batches is reported in EP 5110, and more than 1 batch also in USP 1085. So yeah, that's correct. Well, 3 batches is allowed by FDA. That's actually recommended to test. Well, no, I'm sorry, I mixed up 2 questions. Well, the thing is, if you, as I said, each single assay that you performed is validated during the assay. So each single result has its online validation and is always, we're always sure if this recovery is okay, that we understand, that we are sure that the endotoxins that are that your test method is valid. So this is for most of our customers, it's something we do not do.
Okay, thank you. So the next question is, what about non-endotoxin pyrogens?
Well, according to EP 2630, according to EP 268, I'm sorry, you have to be, well, as I told you, endotoxin are a pyrogen. It's not the only pyrogen, and what you do have to do is to test your first 3 batches. You have to demonstrate that you do not have any other pyrogens present, and only if you have those results, you can rely on product release by performing endotoxin tests. But you have to be aware that well, endotoxins, they are the most potent, or throughout the years, we thought that endotoxins were the most potent pyrogens, but we've come to understand that this is only true in a rabbit, and a rabbit for years has been the gold standard of testing pyrogens. And now with the upcoming of more in vitro assays like the MAT assay, we found out that this is not always the case, that all the other pyrogens can also have a significant effect. And EP 268, which is the Pharmacopoeial guideline for pyrogen testing, it says that you have to test first 3 batches and prove the absence of pyrogens. And if you have pyrogens, you should not only rely on the LAL assay, you should test you should use the MAT assay, because this is actually an assay that also that does not only detects endotoxins. And of course, the same measures then apply, or the same rules apply, as I presented in this webinar.
Thank you. That was really interesting, Peter. So if there's still responses coming in, do you continue to send yours through? So the next question is, can you name further sources of beta glucans, please?
A lot of products from plant origin, they contain beta glucans. I know, for instance, starch, cellulose is a very well-known one. I don't, by heart, I don't know so much more. Cellulose is quite well known. So, but be aware that if you use something from a plant origin, it can be the case that you have beta glucans. Normally, if you're using products from animal origin, that will not be the case.
Okay, thank you. Also, how do you select testing point? How do you select the number of test points?
How do I select the number of test points? And by this, you mean the critical control points? Well, if that is what you're actually, the question is, well, that's something that you need to investigate. You do have to break down your production process. Every single step has to be evaluated. It's probably not always so easy for a non-microbiologist. Well, I'm not saying that it's easy for a microbiologist either, but you have to understand the microbial principles. So each step you need to evaluate, can endotoxins be introduced? If you assume it's not, you have to test this up front. As I said, you have to test your assumptions. So only by breaking down your production process, you will be able to find your testing points.
Thank you, Peter. So another question we have is, how do you investigate the OOS endotoxin? Result, do you know the customer's specification?
How to investigate an OOS endotoxin? Well, when we test for a customer, I think that was the question. Do you know the customer specification? Yes, we do. Well, we have to, and I must be honest. Not all customers think of giving them their specifications. If we do not know the specifications, we do not know that we have an OOS, and we normally do not initiate an investigation. So if you're working with a contract lab, or if your lab is internal, that's something that is really really important, that your testing lab does understand these endotoxin limits, because if you do not have this limit, you lose valuable time, and the longer after the assay is performed, the more difficult it gets to finding a possible source. I do have to tell you that endotoxin contamination during testing is actually the possibility is actually very low, and that's easily understood if you know how much endotoxins that you find in one in one cell, one E. coli cell. If you know what your limit of detection is, well, we're working in a class A laminar flow hood. Even if you would perform environmental monitoring in our lab, we're not working in a classified environment. Of course, the endotoxin is not performed. It's in a controlled environment, but it's not a classified environment. We normally, if we would perform active air sampling in our lab, we found around 10 to 30 CFU per cubic meter of air. Well, you have to understand, you need at least 10,000 up to 20,000 E. coli cells before you actually obtain the limit of an LAL assay. So the likelihood of having a huge contamination in the lab, or it must be that our deperogenation of glassware our oven broke down, but that's something that we normally validate quite often, and that's easily found out. The likelihood of having a contamination in the lab is actually low.
That's really interesting. Thank you very much, Peter. So we have time for a few more questions. So another one is, how widely is HACCP analysis in the pharmaceutical industry?
Well, as I told you, it's the most, well, there are up to two or three different methods that you can use for risk analysis. And HACCP is actually the most frequently used. Brilliant.
So another question is, how many final products should I test from one batch?
As I said, FDA requires, no, I didn't say this, I mixed this up with another question. FDA would say three products, one at the beginning of each batch, one in the middle, one at the end. For medical devices, it's depending, you can find this in ST72, and it depends on the batch size, which can be, if you have a very small batch, below 30 samples per batch, it's 2 samples, but it can go up to 10 samples per batch.
Thank you very much. Also, should an environmental monitoring during the filling process be included as a CCP?
Well, no. It's as I said, you have a very small likelihood of, well, to start with, environmental monitoring is certainly a critical control point for bioburden or for sterility. But as I said, the possibility of this causing a huge effect on the endotoxins is actually low. So environmental monitoring is certainly not a critical control point for endotoxins, no.
Okay, how do you validate the test?
I assume, well, there are different things, the things that you need to keep in mind. As I said, the first thing, and this is something that everybody does, is you're going to assess your inhibition and enhancement. Let's leave out the question, should I do this up front? Should I do this immediately? Should I do this on 3 batches? Well, as I said, this is something you definitely need to assess. We are performing the mostly used method at our lab is LAL chromogenic, which is a kinetic quantitative method. So we always assess this. Is there inhibition? Is there an enhancement? Of course, there are 2 other interfering factors like beta-glucans. If you find out, if you would test your first batches and you would find out that even if you feel like you're controlling endotoxin, you keep on finding high values, there are ways to find out. If you have a trained microbiologist, there are ways to have at least to be suspicious about or to notice that there might be a suspicion of the presence of beta-glucans. So if you find out higher values, that's definitely something you need to assess. Test this by adding beta-glucan blocker, which works very well, or test this by using the recombinant factor C. Also something that FDA says that you have to do, it's also in the same document, in the question and answer from 2012, FDA actually wants you to validate the whole time. So, and I have to be honest, not a lot of our customers actually do this, but it's being, look up the question and answer of FDA. There is actually a part that handles to perform whole time studies. So these validations normally should also always be done or should be taken into account.
Thank you very much. So that is all we have time for today, unfortunately. I would like to extend my thanks to Peter Cornelius for such a fantastic presentation and discussion today. I'd also like to take a moment to thank our audience for taking the time to join us, as well as for submitting all of your questions. A quick note to our audience: as you leave this webinar, a very short survey will appear on your screen asking you to rate your experience today. Please do take a moment to provide your feedback. And if now's not a good time, the survey will also be sent to you shortly afterwards by email. If you could please complete this, it would be greatly appreciated. However, for now, on behalf of European Pharmaceutical Review and Nelson Labs, I would like to once again thank Peter for his time today, and to thank you all for attending today's webinar. We hope to see you again soon. Thank you and goodbye.